Good afternoon, ladies and gentlemen. It is my great honor to serve as today's Master of Ceremonies, and welcome to the 惠蓀 Lectures. 各位贵宾、各位师长以及同学，大家好！很荣幸担任今日活动的司仪，并诚挚地欢迎各位莅临本次的惠蓀讲座。Before we begin, please allow me to introduce our distinguished guests today. 在讲座开始之前，请容我向各位介绍今日与会的贵宾，也请各位给予热情的掌声回应。First, our distinguished guest, the Fellow of the Australian Academy of Science and the Royal Society, Professor David Craik. 首先介绍今日的讲者，澳洲科学院院士、英国皇家学院院士 David Craik 教授。Dean of the Research and Development, Professor 宋正明，研发长宋正明教授。We would also like to welcome our organizers, Director of the Biotechnology Center, Professor 侯明宏，生物科技发展中心主任侯明宏教授。Dean of College of Life Sciences, Professor 黄介成，生命科学院院长黄介成教授。Department of Life Sciences, Professor 林鹤，生命科学系林鹤教授。As we prepare to start, we have a small tradition to attend to. We'll be honored to have Professor Craik come up on the stage to sign the lecture poster. 在演讲开始前，有请院士上台为本次的演讲海报签名。Thank you, Professor Craik. Please remain on the stage. To express our sincere appreciation, NCHU has prepared the 惠蓀 Crystal Trophy as a small token of our gratitude. 为表达诚挚的谢意，本校特别准备了惠蓀琉璃讲座，致赠给院士。We would also like to invite Professor 宋正明 to present the award. 让我有请掌声，欢迎宋研发长颁发这个惠蓀讲座。Thank you, Professor Song and Professor Craik. We would also like to invite our faculty members to come up on stage for a group photo to mark this wonderful moment together. 让我们有请在场的师长上台一同合照留念。In welcoming Professor Song Zhenming for the opening remarks.
Good afternoon, our distinguished guests and our colleagues and students. It's my great honor to welcome all of you to join today's 惠蓀 lecture. Before we introduce our speaker, Professor David Craik, I would like to share with you the background of this lecture. The 惠蓀lecture is the highest honor of this university academic calendar, and we established this lecture to commemorate our late President Tang Hui-sun. And using this platform, we invite the world's most influential thinker to deliver, to share with their innovative insights. You can say that this is the bridge to link this university and the top mind in the world. For say, like earlier this year, we just host Nobel laureate, Professor Brian Schmidt, and he's also from Australia. So today, we would like to continue this proud tradition by welcoming an extraordinary scholar, Professor David Craik. And now I would like to invite Professor Ho to introduce our speaker. Let's welcome Professor Ho.
Yes. Thanks, Professor David Craik, and our dean, and Professor Song and Professor Lin. Because this lecture is organized by Biotechnology Center, I serve as a director of Biotechnology Center, NCHU. So, and also because my research field works on through structural biology, also interested in drug discovery. So I surveyed your research. Oh, I'm very amazing. I think over not 100, it's 1,000 publications. Yeah, over more than 1,000. So that is very amazing during your research. And so I also need to spend some time to introduce Professor David Craik. Professor Craik, he served as a director of the ARC Center of Excellence for Innovation in Peptide and Protein Science. So, like I mentioned, he is internationally recognized and has also over 1,000 scientific publications. And he is best known as a pioneer in the field of cyclic peptide. So as we know, peptide usually is linear form, but he designed and discovered cyclic peptide. That's very potential for therapeutic potential therapeutic application, also nutraceutical application. And this kind of peptide, cyclic peptide, is quite very stable and can resistant enzyme degradation. So, in recognition of his outstanding contribution, Professor Craik has also elected as a fellow of both the American Academy of Science and the Royal Society, two of the highest honor in the scientific communities. So today he will talk about his outstanding research for cyclic peptide, the application in agriculture and medicine. So let's welcome Professor Craik.
Thank you very much for the very kind introduction, and I feel very, very honoured to be here today and looking forward to discussing with many colleagues. So thank you for everybody that's been involved in the organisation. As you've heard, I'm interested in cyclic peptides, and in particular this family of molecules we discovered 20 years ago, which we call cyclotides. They come from plants, and so we like to discover them from plants from all over the world and do their structures, mainly using NMR spectroscopy, but then use them in applications both in medicine and in agriculture. So that's the sort of the background to our work. I always like to start my lecture though by trying to clarify some confusion over my name, because many years ago I did some consulting work for a German pharmaceutical company, and they paid me a lot of money, I think more than I thought I deserved. And when I saw the contract, it was to Professor Dr. Daniel Craig. So I don't know if everybody knows who Daniel Craig is, but he's the actor that plays James Bond. So I was confused because I couldn't see how they could make that mistake. But I do like a swim, and so maybe not so surprising. But I did notice that James Bond is also interested in cyclic peptides, as you can see from his logo from the James Bond films, and I also like cyclic peptides. So there's some common features, hopefully. Except he earns much more money. He gets $137 million for every movie. I also did want to point out that as well as my job at University of Queensland, I'm director of this centre in Australia called CIPS, Centre for Innovations in Peptide and Protein Science. And our job there is to look at interesting animals and plants from Australia and try to use those peptides for drug design and for agricultural purposes. And I'll talk a little bit about the agricultural aspects today, but we're doing things like looking at the composition of milk from koalas for antimicrobial peptides, try to discover novel molecules from Australia's unique flora and fauna, even looking at molecules that can be used to treat stroke and protect hearts after heart attacks, using the venoms from funnel web spiders that one of my colleagues works on. So that centre is based in 6 universities across Australia. It's headquartered at the University of Queensland, but some of you will recognise the Australian peptide scientists that are involved in that centre, and we have many partners around the world, and part of our mission here is to increase our collaborations with Taiwan to have more partners. So we're very welcome to new collaborations, and I'll talk a bit more about the end of the talk. But getting onto the cyclotides, these are cyclic peptides. I first came to notice or to know about them when I heard reports of a Norwegian doctor going to Africa in the 1960s on a Red Cross relief effort. But when he was there, he noticed that when women went into labour in the local hospitals, they gave birth very quickly. And it was because they were making a tea from this plant, Oldenlandia affinis, from the Rubiaceae or coffee family, and sipping it during the labour, and it would accelerate the childbirth. And this doctor, he was a physician, but he wrote a PhD up on the subject of the women using this medicine, and he discovered that the active ingredient was a peptide which he called Kalata B1. In the local language, Kalata means fast. And this is the front page of his PhD thesis, symbolically showing some women giving some of this plant to a lady that's just had a fairly large baby, so maybe she was pleased that the birth was over with very quickly. But he knew that the active ingredient was a peptide, but he didn't know what the structure was. And then 20 years later, I read about this story, and then we did a structure of this molecule, which I've shown here, a cyclic backbone with 3 disulfide bonds, and those disulfide bonds, if I just twist up into this shape here, where 2 of the disulfide bonds form a ring that's then threaded by the 3rd disulfide bond. This was completely unknown at the time. People actually didn't believe me, thought I was crazy. This was the first NMR structure I'd done. But we realised that this structure made the molecule very stable, and that's why women could boil the peptide in the tea and then orally ingest it. Most peptides won't survive that. So I think everybody would know the most famous peptide in the world is insulin, and even after 100 years of insulin as a medicine, we still don't have an insulin tablet. If there was such a thing, it would get digested before it got into the bloodstream. So that's still a problem with peptide-based drugs, to make them orally active and stable. But the plant had already done this already, so we thought this might be an interesting project for drug design applications. And since then, we've discovered it's not just this one molecule, but there's a whole family of molecules which we now call cyclotides. And they occur in all parts of plants, in the flowers, in the stems, in the roots, in the leaves. And so one plant can have dozens to hundreds of cyclotides. And everybody asks, why do plants make them? And they're there as defence molecules to protect a plant. And the analogy I always like to make is, if an animal is attacked by a predator, it can run away or fight or kick or scream, but a plant can't do that. So a plant has to have a very good chemical defence, and cyclotides are the chemical defence against insects. And we proved that many years ago now by taking this Kalata molecule from the African plant and feeding it in feeding trials against a major pest on cotton plants in Australia, the cotton budworm. Cotton doesn't have cyclotides in it, so it has no protection against this insect. And what we did is fed the larvae from this insect with a cotton leaf diet for 16 days, and you see the caterpillar grows to about 300 milligrams in size. But if the diet contains a small amount of cyclotides in it, then the caterpillar doesn't grow or it dies. And I've shown 3 controls and 3 cyclotide-fed caterpillars here. So this is a way that the plants use to protect themselves, and we can put cyclotides onto other plants to protect them. And so we worked with an Australian company called Innovate Ag that was working on another plant called Clitoria ternatia, or butterfly pea, and this naturally produces cyclotides. So what this company did is make extracts from this natural plant and essentially bottle it and spray it from aeroplanes and tractors onto cotton plants, and it's now been approved as a commercial product in Australia and around the world for cotton and vegetable crops, and it's been a very successful product. The advantage of it is that it's not toxic to beneficial pollinating insects. So one of the hazards of traditional insecticides is that they can cause damage to beneficial pollinating insects like bees, but this is completely safe and it's been approved for use on a wide range of crops now. So we were very pleased that this has sort of reached the market now, and the active ingredients are cyclotides. And how does it work? Well, if we look at the 3-dimensional structure of a cyclotide, it contains some green residues here that are the hydrophobic or the greasy residues, and we know that they'll be attracted to membranes. So we always thought that this would act by binding to membranes in the guts of caterpillars. And this is a normal caterpillar that hasn't been eating cyclotides, and this is the gut of a caterpillar that's been eating cyclotides. And you can see it disrupts the membranes, stops the feeding behaviour, or kills the insect. And for about the last 15 years, we've been doing a range of studies to try and understand the physics, the biophysics behind exactly how this molecule works. And I can't talk about all of those studies, but I did want to say that peptide chemistry has been very important because we've now developed ways of making these molecules and understanding how they work. And the students and postdocs that have done this work are shown in the slides. But this is the Calata molecule using the one-letter amino acid code, showing the cysteine knot in a schematic form. And what we did is what is typical for peptide chemists to do an alanine scan or a lysine scan, where you change every residue one at a time to alanine. And if we replace this residue here with alanine and the molecule loses activity, then we know that must have been an important residue. And it turns out that these 6 residues here are very important for the insecticidal activity. If we replace any one of them with alanine, we completely lose the insecticidal activity. So I always ask the students in the audience if they can tell me why those 6 residues would be, you would think that they're important, because they're spread everywhere across the sequence. They're positively charged, negatively charged, polar, and all very different amino acids. And so it's not obvious when you just look at the sequence. But when you look at the structure, you see that they're all located on one face of the molecule, which we call the bioactive face, and they're centred around this glutamic acid residue. E is the code for glutamic acid. And so this is the bioactive face of the molecule and is obviously important for activity. This is the hydrophobic face, which is in a slightly different spot, but it's also important for activity. And if we flip the molecule around onto the other side and look at the back of it, we also changed every residue one at a time to lysine, and we found that these blue residues here, if you change those to a lysine, you can make the molecule a better insecticide. So we can actually improve upon nature. We can fast-track evolution by designing improved versions of what nature's produced, and that's one of the aspects of the research we're doing. So we had now used chemistry to understand how this molecule, where the active parts of the molecule are, and it highlighted to us that this molecule was very tolerant to mutagenesis, to changing the sequence, to synthesising it, and that was very useful for pharmaceutical applications later on. But to understand the insecticidal activity, we noted that this glutamic acid is completely conserved in every cyclotide we discovered, apart from one, and that was a very similar amino acid, aspartic acid. And what happens is the negative charge on this amino acid gets attracted to the positive charge on phosphatidylethanolamine lipid head groups and attracts the molecule to the membrane, and then the hydrophobic patch helps absorb the molecule into the membrane and disrupt the membrane. So it's simple attraction using charges that gets this molecule to membranes, and as I said, we've now have a good understanding of how these molecules work as insecticides. But as I said, the work also allowed us to explore other aspects, and one molecule I'll come back to later is so-called T20K, where we change this threonine to a lysine, and that molecule has now been in clinical trials as a drug for multiple sclerosis, and I'll talk a bit about that later on. And other examples that came out of the lysine scan work have now been patented with Syngenta, a big ag biotech company, for other insecticidal applications. But I'll just finish the agricultural side of things by saying just in the last few months, this product has now become commonplace in Australia in every hardware store across the country for the home garden market. It's labelled as being safe for bees, and so the home gardener can now kill their pests in the garden with safety for not damaging dogs and cats and beneficial pollinating insects. So we're very excited to see some basic peptide chemistry now get into the everyday marketplace. And I just on Sunday in Australia, there was a national ABC television show talking about the discovery of CeroX. So I do have some links with James Bond with the movie star role, but not a very strong link. But this story went to air a few days ago, and so now we think that this area will get the public more interested in peptides as eco-friendly pesticides. So I won't talk more about the agricultural applications now. I'll move on to the pharmaceutical applications. But just to say that the agricultural applications, we look at the natural activity of the cyclotides and we use them to protect crops. With the pharmaceutical applications, what we're doing is taking the stable framework and saying we could engineer that framework to make it useful to stabilise peptide-based drugs. And the rationale for doing that is, here's the topology of cyclotides and the topography, the 3D shape, and between the cysteines we say that there are loops, backbone loops 1, 2, 3, 4, 5 and 6, and they stick out from this core and get exposed, and we can use them to interact with other biological targets. So we know from the natural cyclotides that every amino acid between the loops is very variable, except for the cysteines and one or two other residues. So we can conclude that we could vary some of those residues with a design sequence that might have a therapeutic application, like an anti-cancer drug or a drug for cardiovascular disease. So we're actually making designer cyclotides, and we call this molecular grafting, where for the gardeners in the audience, it's the same as what you do with gardening. You take a rootstock and you put another plant onto it and make a new plant. In molecular grafting, we take our rootstock of our superstable cyclotide and graft other elements of structure on that will give it a new biological activity, preferably a therapeutic one. My favourite example of a grafted plant is this one here, which is a graft between a cherry tomato and a potato. This is real. You can buy this on the internet for €19. They're called pomatos or tomtatos. So I like them because you can have your French fries and your tomato ketchup all on from the same plant. But when we do molecular grafting, what we actually do is use chemistry to make these molecules. I won't go through all the details, but basically we attach our peptide, our growing peptide chain, to a resin and assemble the amino acid sequence so that we have a cysteine at the N-terminus, and then there's a series of reactions which is called native chemical ligation, invented by Steve Kent and Phil Dawson, that allows us to make these cyclotides quite efficiently in the lab so that we can now make our own designer cyclotides with a desired sequence. And we started making the native molecules and doing alanine scans, but now what we wanted to do is make designer ones for therapeutic applications. So the rationale is we've got our desired stable framework. Someone out in the literature or in a pharmaceutical company might discover a peptide, it might be a 6 amino acid peptide, that's potent for activity against a particular disease. And the diseases I've shown here are all ones that we've worked on so far. But we know that linear peptide will not work as a drug because it would get broken down by proteases, just like insulin gets broken down if there was a tablet. And so it would never make it as a drug. But what we proposed is that we could graft that framework onto a cyclotide and it would inherit the stability properties, and that turns out to be true. We can really guarantee stability of any small peptide epitope, and with luck we can maintain the biological activity of the epitope and then have a stabilised therapeutic. And we do this with lots of different types of cyclotides. There are 3 main families. I've shown 2 of them here, so-called Colada type or the Moebius type and the trypsin inhibitor type. I'll come back to these later. But they have natural variation in the sizes of these loops. And what we do is we choose one of these frameworks and we graft peptide sequences anywhere between 1 and 21 amino acids is what we've done so far and what other people have done in the literature, into one of the loops. And just to illustrate 3 examples, this is typical of what we would graft in. This is a polyarginine sequence. This was discovered by another group as having the ability to stop the growth of blood vessels in tumours. That stops the tumour from growing. This other sequence here had activity against a receptor involved in appetite, and there was another one here involved in lung cancer. So what we do is we graft those sequences into one of the loops based on what we know about the structures, and we do a little bit of trial and error and can graft into one of several loops. If that doesn't work, we can go into another cyclotide framework that has slightly larger loops. But to cut a long story short, we've done grafts of every amino acid sequence from 1 to 21 amino acids in many, many of these loops, and we very often have success in producing a bioactive molecule that's now super stabilised. So that's the principle, and the loop structures can, or the epitopes can have a whole range of different sequences, so it's a very versatile platform. And I'll just illustrate it with some early work in the area of obesity, where I live in Queensland here, the obesity rates have been rising over recent years. Our diets aren't getting any better. We're eating too many French fries and not exercising enough. Maybe the pomato won't solve the problem either if we start eating that, but also it's not going to be good for the acting career if we keep letting obesity rates go up. So we're interested in therapeutic interventions in obesity. And at the time we started this work 10 or 12 years ago, the melanocortin receptor was a major pharmaceutical target for obesity. It's a receptor that controls appetite in the brain, and there are natural hormones, and people knew that 4 amino acids were the vital part of some of these hormones. So what we did is graft that tetrapeptide sequence into a cyclotide framework and showed that that molecule specifically bound to the melanocortin 4 receptor with a binding affinity of 29 nanomolar, which is quite potent, not quite good enough to make it a drug in itself yet, but a good starting point. And then what we did is further optimise things by taking a smaller cyclic peptide framework of 14 amino acids with 1 disulfide bond and doing the same thing, grafting this tetrapeptide epitope into there. And because this was a smaller peptide, we were able to do more sophisticated chemistry and do N-methylations and D-amino acids. And we've been working with AstraZeneca on further developing this as a probe for melanocortin receptors. We got down to about a 2 nanomolar binder here, and as I said, we're still working on this. But in the meantime, of course, the whole area of obesity has been taken over by GLP-1 agonists, molecules like Ozempic that I'm sure you've all heard of. They attack a completely different target, but they're peptides, quite different peptides, but they're really revolutionising the treatment of diabetes and obesity in medicine. And so we think that this melanocortin work will have a niche place as a sort of an adjunct on the side of some of that GLP-1 work. The second example I'd like to give you is one of the other major targets in pharmaceutical research is getting drugs inside cells, particularly for cancer targets. And it turns out that some cyclotides can actually penetrate into cells and deliver cargoes. Most peptides can't get inside cells. Antibodies can't get inside cells. So, and big proteins can't get inside cells. But cyclotides, some of them can, and so they can be used to attack important targets for cancer. And the example I give comes from this tropical fruit, Momordica cochinchiensis. A French-Vietnamese group discovered the peptide from this fruit in 1999. We did the structure in 2000, and then 10 years later discovered that it could actually penetrate cells. And we've been working on this framework ever since. The original work started with a field trip up to the far north of Australia at near Cape York here. Brisbane's here, so there's 3,000 kilometres of distance between, and once you get to Cairns, there's no more roads, it's just bush tracks and rivers that don't have any bridges, so you have to drive through the rivers. So it was quite fun to go on this field trip and collect this plant, and this is me here and the PhD student here, Shane, trying to collect some of this fruit from the rainforest. But to get there, we had to cross rivers in our four-wheel drive. But of course, before you can drive through the river, you need to know how deep the water is. But it's always a problem because there's crocodiles in the water. So my students decided I should be the one that went through the river. So I've managed to survive without losing any legs. But we did manage to collect this fruit and do the structures and do the work. But then we discovered that you can buy this fruit anywhere in any market in Thailand. So it's become much easier to get hold of this fruit. And it's well known in Southeast Asia. I'm not sure whether it's known here in Taiwan very well, but it's a very famous fruit that's used for colouring sticky rice at weddings in Vietnam, for example. But the peptide comes from the seeds of this fruit, and it's a cyclotide. So what my colleague Julio Camamero did is had a sequence that had been discovered as a binder to a protein-protein interaction inside cells called P53HDM2, and he took this sequence that had been discovered from phage display and grafted it into a cyclotide framework, as I've schematically illustrated here, and showed that that was active in a mouse tumour model. So this is a mouse with a tumour implanted. The tumour will just keep growing as the blood vessels develop, but if you measure the growth with no treatment, then the tumour just keeps growing. The best molecule known at the time is in green here. It was slowing the growth down a little bit, but you can see the cyclotide was much more effective at reducing the tumour growth. So from very early studies we could show that we could get a peptide inside cells and stop the growth of tumours. So this was the start of this work. We've continued in this area and are optimising the molecule. I won't go through all the details. It was recently published, but basically what we're doing now is grafting onto two loops of the cyclotide framework, one where we put the binding sequence or a range of different binding sequences, and the other where we put on an additional cell-penetrating peptide to try and get the molecule inside cells. And this just as an illustration from the table of contents graphic showing this cyclotide with a fluorescent dye getting into the cytosol of cells and interacting with this target. Just to summarise that this is an approach to blocking a P53. P53 is a very famous cancer protein involved in so-called guardian of the genome. And as I said, the cell biology shows that the molecule very nicely gets up into cells. And the bottom line that I'd like you to remember is that we can now show with cyclotides that we can get peptides inside cells, which was previously unachievable. So that's where we were up to until about 3 or 4 years ago, when we were designing molecules one by one. When a new target was discovered, we would design a cyclotide against the target. But then we started to get interested in display technology, where rather than making the molecules one at a time, we could make a library of a million molecules, 10 to the 12 molecules, using so-called mRNA display or rapid technology that was invented by Professor Hiroaki Suka from University of Tokyo in Japan, who's now been a very famous scientist that created a company called PeptiDream, which is a $6 billion company that uses this technology in drug design applications. So we collaborated with Hirosuga to look at a target that's involved in a life-saving treatment called ECMO to design an inhibitor of an enzyme called factor 12A. And just as an aside, I should say this is Hirosuga. He's a very famous peptide chemist, but also a very famous guitar player, and he's used his profits from PeptiDream to buy many very special guitars. And so we've had him as our conference musician for a conference we organised a few years ago. But what we were really trying to do here is inhibit this enzyme factor 12, which is implicated in clotting disorders, in a treatment called ECMO, which is extracorporeal membrane oxygenation. So when patients are very sick in hospital and they need to be on life support, their blood supply is redirected through an oxygenation machine, but that involves the blood going through tubing, and that tubing causes clotting. And so surgeons try to stop the clotting by providing heparin, but then that causes bleeding. So it's often a very delicate balance between the two. And we were trying to design a molecule that would be better than heparin in this approach. And so we used this mRNA display to vary the loops, as I said, 10 to the 12 variants put into these loops using this display technology, I think, which is pretty well known now, so I won't go through the details. But basically you can have your peptide displayed linked to a barcode that allows you to fish out that one molecule in 10 to the 12 that has activity against your target. And this is typically what we did. We had our parent peptide. We varied all the amino acids combinatorial in loop 1 and loop 5. As I said, 10 to the 12 combinations. You make the library, express it, in vivo translate, and then pan it against your target, fish out using the barcode the desired molecules, and repeat the process, and eventually you enrich and get some sequences that are optimised in the library, and you can then look at those sequences and say these would be interesting molecules to chemically synthesise. So, as I said, this really accelerates things many fold, and you get hits much quicker. What we then did is synthesised a collection of those hits and made the molecules in large enough quantities to test, and found that one of them was a 37 picomolar binder to this enzyme factor 12, which is exactly what we wanted to get. So a very potent binder, and also very selective in hitting just that enzyme and no other enzyme that was related. And so we call this molecule various names over the years, but FX1 here, it's very stable in human plasma. A normal peptide degrades within a few minutes, but this was stable over 24 hours. Well, actually, a series of them were very stable. We can do the structures by NMR and tell that the molecule has identical folding to the native scaffold. We can test it against the enzyme we want, and it's very potent, but it's very weak against all the other enzymes in the coagulant cascade, so very selective. And we can show that it increases the clotting time, so it stops the blood from clotting too fast. So we're very excited about this, and eventually we're in the stage now where we're trying to raise funds to commercialise this so that it could eventually go into patients who are on these ECMO machines, which you can see the tubing with the blood going into the patient, being recirculated and oxygenated. So we haven't put it into people yet, but what we have done is do a mock-up ECMO machine that has all the exact parts of the machine that the patient would use, using human blood and circulating it and looking to see that the blood doesn't coagulate. We published that just late last year, a few months ago, and we're looking forward to taking this forward as we go forward. The molecule that we've developed is actually better than heparin in every one of the hemodynamic parameters that we've measured. So it's looking quite promising, but there's still some way to go before this goes into humans. So that's a couple of illustrations. There's many more examples now from our group and others where this cyclotide framework has been used against enzymes like the factor 12 I just mentioned, like protein-protein interactions, and like cell surface receptors like the melanocortin receptor. But the big thing that we're doing now is how do we make these molecules cheaply and efficiently and sustainably so that when we go into production of hundreds of kilograms and tons of peptide, that it can be done sustainably. So for that we had to understand how plants make these molecules, and to cut a long story short, plants express a precursor protein that contains a cyclotide domain flanked by an N-terminal region and a C-terminal region, and I won't go through the details, but there's a successive series of cleavage steps that releases a small peptide whereby a glycine interacts with an asparagine and makes the cyclic peptide. And it's an enzyme called an asparagine endopeptidase that does that. Every plant has at least 2 or 3 or 4 asparagine endopeptidases, normally to cleave after asparagine, but some plants have evolved to do this transpeptidation or the cyclisation reaction, and they've stumbled across the fact that when you make cyclotides, they're very good defence molecules and very, very stable. So having understood the biology and the synthesis of these molecules, we can now apply that to make designer cyclotides, and the concept is very simple. We know that plants are full of DNA. That DNA encodes peptides. We come along and say we don't like those peptides, we want to redesign them as drugs, so we reverse translate, make a modified gene with a new piece of DNA in the gene. We can then put that back into a crop plant, and that plant will now produce the cyclotide that we want, not the one that it wants. And we could then express that cyclotide in the leaf, and I've shown soybean here, and harvest the plant, and now we've produced a pharmaceutical in an outdoor factory in the field with just a bit of sunlight and a bit of water and a bit of land, and that can then become a pharmaceutical that's produced very safely and greenly. Just for relevance, Ozempic is causing a worldwide concern in terms of sustainability. It's now known that 1 kilogram of an active pharmaceutical ingredient produces up to 10 tonnes of waste, or between 3 and 13 tonnes of waste products and solvents. And so the pharmaceutical industry is very concerned to make greener ways of producing peptides. So I'll tell you briefly about that work, but also what we're doing with now thinking about could we actually express a peptide in a part of a plant you could eat, like a tomato, which I've shown here, and then your medicine becomes your food. So I'm a little bit behind the times here because 3,000 years ago, Hippocrates said that let food be thy medicine. And so now we're finally getting to the stage where we might be able to do that. And we're doing it with lots of little model plants. Arabidopsis is a test plant, the tobacco I mentioned, or I will mention the tobacco, but a whole range of other plants, and looking at a range of different diseases, including the multiple sclerosis example that I mentioned earlier. And we're doing it with a whole range of different peptides that we've discovered in our group, from the cyclotides to molecules from scorpions to molecules from cone shells that are potent painkillers, through to sunflower molecules. And we're all putting them all into plant hosts as ways of producing them cheaply. The reason we choose plants is because all the lab work is done with peptides, and it's efficient to do this synthetically for small amounts, for milligram amounts of peptides in the lab. But when you want to get onto the ton scale, you need either recombinant approaches in fermentation facilities, which can be very expensive to maintain, or we're now going to plants which are sustainable, very expandable and scalable. And the preferred plant we're using is this native Australian tobacco, Nicotiana benthamiana. It was a plant that some of you might know was approved for producing antibodies for the Ebola virus in the 2014 outbreak. More recently, it was actually used by Medicago, a Canadian company, for making a COVID-19 vaccine in Canada. So there's precedent for people making antibodies, vaccines in plants, but no one else in the world, apart from us, is doing it with peptides. And the concept is we know the plants that make cyclotides, we know that they have the precursor proteins, we know that they have asparaginal endopeptidases, some of which are proteases and some of which are ligases, and it's the ligases we want because they produce mainly the cyclic peptides. So when we started, we originally thought, well, let's just put the gene for a cyclotide into a non-producer plant and see if that works. And it did to some extent. We produced a little bit of cyclic peptide, but not very much, and we had a lot of linear peptide there. But when we also transfected the plant with an AEP that was designed to cyclise, then we got increased yields of the cyclic peptide and decreased yields of unwanted products. We also went on to modify the plant further with CRISPR technology to edit out some of the unwanted proteases so that we could increase our yields. I won't go through all the technical details of that, but we're doing that with gene editing in a range of different plants with the aim of making plants optimised as factories for making pharmaceuticals. First example we published was on a plasmin inhibitor that was a 25 picomolar inhibitor of an enzyme involved again in the blood coagulation cascade, and we showed that peptide was identical to the synthetic one. The plant-produced one was identical to the synthetic one and was super active. We then went on to take a peptide that we discovered from a marine cone snail that's 150 times more potent than morphine as a painkiller, and originally showed that in normal tobacco we couldn't produce that peptide, but if we had the engineered tobacco plant, then we could produce some of the cyclic peptide, and we're optimising that at the moment and playing around with all the molecular biology to try and optimise that process. We're working with the US Department of Defence to make anti-malarial peptides in plants, and that work's been going on for a couple of years and looking very promising. And we're, as I said, engineering these plants to make the native tobacco shorter and have a higher biomass so that we can then use it in robotic-driven factories where it's more optimised for manufacture. This is a company we're working with in Sydney in Australia that grows spinach for the supermarket industry. Each of these racks, so there's 1, 2, 3, 4, 5, 6, about 10 rows of racks with spinach plants growing under artificial light inside a factory, powered by photovoltaic cells and using robots that scan across each plant once every hour and measure how much it's grown, photograph it, how much it's grown, how much water it needs, and uses AI to optimise the growth. And we're now adapting that technology to grow these tobacco plants that will make these pharmaceutical peptides with applications for pain and cholesterol and obesity. So that's work in progress. Just one example of how we're getting ready for the multiple sclerosis drug. We're now producing that in tobacco plants and extracting it and showing that it's identical to synthetically produced peptides, so we can produce it cheaply in tobacco plants. And now Lynn has even gone ahead and put this into tomato plants, and this shows a mouldy image of a regular tomato with a transgenic tomato that's producing this multiple sclerosis drug. Not in all tissues so far, but we're optimising this. So down the track, it actually will become a reality for food to become medicine in the case of this multiple sclerosis drug. The principle would be someone with multiple sclerosis could relieve their symptoms by having a tomato a day, or having some tomato juice in the morning for breakfast, or having a Bloody Mary with their evening drink to maybe cure the multiple sclerosis symptoms. So all sorts of new ways of thinking about food and medicine as we go forward. So I'll wind up there by saying that we're now working on this analgesic peptide that's more potent than morphine. We're looking on the multiple sclerosis example. We're looking at surgical applications. I didn't mention prostate cancer, and we were in the process of making transgenic potatoes to put that anti-obesity medicine into. So I'll wind up there by saying what I hope I've shown you is that we've gone from discovering peptides in plants, characterising their structures, deciphering their functions, and then re-engineering them, and now we're actually completing the circle by putting the peptides back into plants as production factories. So that sort of brings the analogy with 007 back as a circular sort of argument, and I think you all know that 007 is licensed to kill. So we're not licensed to kill, we're licensed to save people, but we're licensed to pill as we go forward. So I'll just finish there, but I wanted to spend 1 minute at the end by saying that this is the sort of work that's going on in our Centre of Excellence, the SIPs that I mentioned, and we are very much looking forward to looking for collaborations with scientists from around the world, and here in Taiwan especially. Annie, who's in the front here, is the Manager of International Connections, and we've got her details here if people wanted to connect and look at possible collaborative opportunities and exchange visits to Australian labs. And the QR code you can scan and get the details. But just to highlight the way we think that we're doing blue sky research. So here's Annie at the American Peptide Conference looking at the blue sky, thinking about how we can dream up new ways of using peptides. But on the back of her T-shirt is our motto, unleashing the power of peptides and proteins for human benefit. So that's where we're at. I mentioned the people that have done the work as I went through the slides, but I just wanted to say that the other thing that we're doing is organising a summer school in peptide science in July on an island in the Great Barrier Reef. This is available for students around the world to apply and come to. There is a cost to coming, but it's very reasonable for 7 days accommodation on the island and all the tuition fees. And you can apply by linking up with Annie or through the QR code. You can go swimming with green turtles on the island and learn about peptide chemistry from some of the leading scientists in the world. So I mentioned Hirosuga, but we have really some of the most world-renowned scientists coming on to interact one-on-one with students. It'll be limited to about 40 students across the world, so we're excited about organising that. So feel free to explore the website if you are interested. So I'll finish there, but by saying that I hope I've given you some impression of what we're doing in terms of peptides in medicine and agriculture. Thank you again for the wonderful hosting so far in Taiwan, and it's an honour to present this lecture, and I'm happy to answer any questions if there are any. So thank you.
Okay, very, very wonderful talk. I also interested in your design, how to design the cyclotide. But now we open a question for the audience. Any questions? I'm interested because your cyclopeptide is produced by plant or use a synthetic method to synthesize the cyclic peptide? I mean, most of the cyclic peptide you show here, you synthesize this cyclic peptide by plant or...
So we originally discover them in plants, so we isolate them from plants. Once we characterise their sequences, then we tend to synthesise them in the lab using peptide chemistry, or now using the plants to make them.
Yeah. Also, I mean, also you use some like the drug conjugate with your cyclic peptide together, and also have some potential usage for anti-cancer or something. I just interesting how you conjugate the compound with your cyclic peptide together.
Yeah. So I didn't talk about that work today, but that's something we're doing with the malarial area, putting anti-malarial drugs onto other molecules to help carry them into cells. So that's just using classical maleimide chemistry to link the peptide with the drug conjugate. So we're not inventing any new technology to doing that. That's quite well-known technology for linking drugs onto peptides.
Yeah. Because my expertise was on to develop some DNA anti-cancer drug. Whether it's possible to conjugate our compound with your cyclic peptide?
Yeah. So it's something we haven't done. Peptide nucleic acid conjugates so far, but that's certainly possible and something that we're thinking about for the future. And I think that's a very promising area because the peptide is very stable and can carry the nucleic acid to a cell and even take it inside the cell. So I think that'll be a really important area for the future.
Yeah, okay, thank you. Any questions?
Yeah. Well, thank you for your very interesting talk. And I have a couple of questions. One is that from bacteria, or is there any production for this kind of cyclic peptides? And the second question is that I noticed that there's a cysteine inside, but is there any interaction with the metals or heavy metals for that?
Yeah, so both good questions.
And the third question would be is there any mechanism that already know that how the cell uptake this kind of
Yep. I have to remember all three questions. So the first one, bacteria. Yes, bacteria do produce cyclic peptides. They tend not to produce them with disulfide bonds, but there are several classes of cyclic peptides that bacteria produce. And as you know, when nutrients get scarce, bacteria secrete all sorts of molecules to kill their neighbours so that they can get the resources. And there are bacteriocins. There are some cyclic peptides that range in size from about 50 amino acids to 78 amino acids that fold up, that have a head-to-tail cyclic backbone and helical structures, and they're quite potent antibacterials. One of them we've proposed might be useful in the food industry as an alternative to niacin, which is a very widely used preservative. So we think that that's an underexplored area, and the reason it's underexplored is cyclic peptides are a little bit hard to see using mass spectrometry. They don't fly very well on mass spec. So proteomics people tend not to discover them very much. And genome people don't discover them because it's hard to know whether something will be cyclic unless you know the cyclisation mechanism. So to discover new classes in bacteria is a little bit problematic, but I think it's with new AI and bioinformatics areas, then that'll be very important. So yes, bacteria definitely produce interesting molecules. Second question on the cysteines, yeah, that's a really insightful question because we always thought that maybe metals would bind. And so we tested many, many metals to see, and we haven't yet found a strong binding interaction in the centre, but it's really interesting, and I think there's a lot of potential because plants that produce cyclotides are often some of the first that colonise mine sites. So when there's a site with toxic metals around, some of the viola plants that colonise first are actually cyclotide producers. So I think there is something in there, and I know there are groups in China that are looking at cyclotide plants as mine remediation plants. So I think there's something there, but we've never been able to see strong interactions between a range of heavy metals and cyclotides, but I'm sure there's something there. And third question was... the uptake. So the uptake is largely endocytic mechanisms. So the Carlata type molecules bind to membranes, as I mentioned, to phosphatidylethanolamine. So they bind to the membrane and then they get engulfed using endocytosis, which means that they're in sacs, endosomes inside the cell. So they're not really inside the cell, they're just inside a sac. But because they're so stable, they can leak out of the endosomes and get into the cytoplasm, and that's why they can interact with a cytoplasmic target. So it's endocytic mechanisms in general for two of the classes, and it's their stability that allows them to get into the cytoplasm. For the third class, the trypsin inhibitors, they actually don't bind to cell membranes, but they get in through macropinocytosis, so just engulfed by the cell, and the same thing, they're stable so that they can get into the cytoplasm. So I think there is some passive diffusion as well for some of them, maybe some receptor-mediated transport, but that's not yet been proven. So often, as scientists, we always think there's one mechanism for something, but usually nature has multiple redundant mechanisms, so I think there's probably several ways, but endocytosis is the main way. We not naturally, but one of the things you could do is put in a nuclear targeting signal. One of the things we like about these molecules, they've got 6 loops, so you could put in a signal to target to a cell type, like to a cancer cell. You could then put a cargo on, and then you could put a nuclear targeting signal, or to some other organelle within a cell to try and target. So that's the work of another colleague of mine in the centre, Sonia Enriques, is doing a lot of. Yes, yeah.
Oh, thank you for your speech, Professor Craik. And it's such a great honor to have this chance to learn from you and your study. And I'm curious about the application. You've mentioned like one of the application of the cyclotide as an agonist on the receptor in the brain. So it makes me wonder, like, does the cyclotide naturally permeable to the blood-brain barrier, or do we need to design any specific sequence on it to graph it and to achieve the central nervous system delivery? Thank you very much.
Yeah, no, thanks for the question. So it was a bit of an echo, but you were asking about the blood-brain barrier, and the answer is cyclotides don't penetrate the blood-brain barrier in a normal animal or human very well, like most peptides. You know, our brain is designed to keep out stuff like peptides, but under conditions like multiple sclerosis, the blood-brain barrier is compromised, and under cancer conditions as well. So under those conditions, there seems to be sufficient penetration of the barrier to get into the targets that are inside the brain or on the border of the blood-brain barrier. So we don't understand a lot of that now, but we think it's where the barrier is compromised, and so diseases that involve that compromise of the blood-brain barrier will be the ones that we could use. We probably can't use cyclotides for other sorts of conditions where that barrier is not compromised so far, unless we learn more about how to penetrate that barrier. And that's one of the grand challenges of the centre we've got, is how to understand how to get peptides through a whole range of barriers. But it's a really good question.
Thank you very much, for Professor Craik. I would like to ask if there is any structural or functional limitation, either known or possible, a limitation that the cyclic peptide can or cannot do, or as long as you know the motif and you can fit it inside the cyclic peptide, and it should be working.
Yeah, so again, good question. So we choose to want molecules that are not any bigger than 50 amino acids. So when we graft into a cyclotide framework, we normally would just put in 6, 10, 12 amino acids so that the total number is around 50, so that we won't necessarily cause unwanted immunological issues. So that's just what we choose to do, but there is actually no real barrier. You could put in a huge peptide sequence into one of the loops of a cyclotide, and technically you could do that, and that's what actually nature does. So in our bodies we have nerve growth factor. It's basically a cyclotide with a cysteine knot in the middle and huge loops on it. So nature's already doing that. It doesn't quite have the stability, because once you have a big loop, floppy loop, it will be subject to proteolysis. But the cysteine knot does have some level of stabilisation. So yeah, it's possible to put bigger loops on, but we don't quite get the same advantages as if we keep it into smaller systems. And just for the other reason that we don't do bigger loops is we don't want to put extra stuff on that will interfere with the binding. We want to try and keep the molecules as small as possible so that the loops don't interfere with non-target binding sites.
Professor, thank you for your speak. I would like to ask you one question. So as I understand, these plants produce the peptides to protect themselves against the insect, right? So I believe also humans as also threat for the plants. So is that possible? These peptides can have some toxic effect on the humans, like human body system?
So far, we haven't seen any toxic effect. If you eat in fancy restaurants and you have viola plants on your salad or in your soup, you're eating cyclotides. They're in every viola plant, which are often used in salads. They're in the tropical fruits that people ingest as colourings in sticky rice. There's even a gin product that is coloured blue by the butterfly pea flower that contains cyclotides. So if you drink ink gin, then you're ingesting cyclotides with no harmful effects. Maybe the alcohol harms you, but the cyclotides won't. So we haven't seen any harmful effects in humans so far, because our gut membranes are different composition to those of the insects that are eating the plants. And so we don't have phosphatidylethanolamine on the outside of our gut membranes the same way that insects do. So far no toxicity, but we won't really know for sure until we get more molecules into human clinical trials. But at the moment we think they're safe. The insecticidal product that I showed you was the first peptide product approved in Australia by the regulatory authority with no upper limit on its use. So it was judged to be so safe that farmers could spray as much as they wanted without harming beneficial animals, dogs and cats, and safe for the environment. So by extrapolation, we think it'll be safe for humans. Of course, farmers won't spray as much because it's expensive. So even though they're allowed to use as much as they want, they would always just use as much as you need.
Yeah, I have a final question because now the GLP-1 is very famous. Yeah. Also, will it possible to use your method to synthesize a cyclic GLP-1?
Yeah, so maybe not such a cyclic one because it's a very long molecule, but we have thought about producing other peptides in plants. GLP-1's a hard one because it has modified amino acids, so it has a lipid tail on it that so far plants can't do, but we could certainly produce the peptide itself and then do a conjugation reaction using our enzymes to conjugate a lipid on, or we're even thinking of re-engineering plants to see if we could do the lipidation, but that's some way up in the future. But I think that's what we've got to do with manufacturing peptide-based drugs, because the regulatory authorities in Europe have banned DMF and some other solvents or regulated them because of the toxicity to the users. So more and more chemical manufacture is going to have to be, toned down and more biological ways of manufacturing things made. So for the future, I think it'd be really neat to do that, because the market, obviously, for Ozempic is huge. It's estimated it'll be a $20 billion seller within the next few years, and the market for GLP agonists, people estimate anywhere between 100 billion and 200 billion by 2030. So I think there'll be a lot of people working in this area.
Thank you. And if no more question, we thank Professor David Craik again and his wonderful talk. Thanks.